All right, we're going to go ahead and get started here with the broadcast.
Hi everyone, and welcome to ASBMB Journal Club. I'm Raj Mukhopadhyay. I'm the science writer for the American Society for Biochemistry and Molecular Biology.
And I'm Jeff Hunt. I'm the Society's Public Outreach Coordinator.
So ASBMB Journal Club is this new interactive feature where we are going to highlight some of the authors who publish with ASBMB journals. The Society publishes 3 journals: Molecular and Cellular Proteomics, the Journal of Lipid Research, and the subject of today's hangout, the Journal of Biological Chemistry.
All right, so there you can see our journals. And today we are honored to have started here.
Yes,
Broadcast. I guess there's an echo. Oh, I know why there's an echo. All right. So like I said, we're honored to have 2 authors from a recent JBC paper of the week, Douglas Thomas and Jason Hickok at the University of Illinois Chicago. So say hello, guys.
Hey.
Hey, everybody out there. Thanks for tuning in.
Glad you guys could have us. We're pretty excited about chatting here today. So.
Yeah, first I want to just thank the American Society for Biochemistry and Molecular Biology and both you two, Jeff and Raj, for putting this all together. And we're really excited to have this opportunity. We hope to get some great questions and some good feedback.
Awesome. And where are we there? So we are going to be talking about their paper of the week, which we have shown up on the screen right now. And so the papers of the week are really the top 2% of the papers that are published in the JBC, and they're selected by the associate editors and editorial board members. So it's really an honor to get this distinction, and these are really the best kinds of papers that are being published in JBC every week.
So the paper that Jason, Doug, and their co-authors had published in the JBC about a week and a half ago now, talks about how nitric oxide can affect histomethylation. So the idea that they put forward in their paper is that a molecule like nitric oxide can affect epigenetics and bypass the classical cell signaling mechanisms that we traditionally think about when we talk about gene regulation.
Right. So we are going to go, and we'll start off asking some questions here, but people who are watching, and feel free to join in and ask your own questions. We want this to be a discussion and not just sort of a back and forth between us. If you have questions, you can put them on our Google Plus page or tweet us on Twitter. We're at ASBMB, and just use the hashtag Journal Club, and we'll try to get those questions answered. So let's get started. You guys ready?
Yeah, let's do this thing.
All right, folks, sorry about that. We were having some technical difficulties, but it looks like we managed to get the 2 most important people on this hangout, which are Douglas Thomas and Jason Hickok, who are the authors of the paper of which we are trying to discuss. So, so I think we're going to cut the intro and just go straight into talking about the paper. The paper we're talking about is a JBC paper that Doug and Jason had published in the Journal of Biological Chemistry about 2 weeks ago, and the paper talks about how nitric oxide could affect histomethylation. And the paper actually just puts forward the idea that a molecule like nitric oxide can actually affect epigenetics and bypasses the classical cell signaling mechanisms that we normally think about when we talk about gene regulation. So Doug and Jason, I think I'll let you take it from here. And folks, if you have questions, please tweet them to us at ASBMB. That's our handle, and the hashtag is Journal Club.
All right, thanks a lot, Raj. Jason, can you work the screenshots? I'm on a different computer here now.
Yeah, no problem. I'll take care of it.
I don't have that on here. So anyways, thanks a lot. We want to briefly thank the Society for Biochemistry and Molecular Biology, and again, thank you, Raj and Jeff, for setting this whole thing up. We're really excited to be here to discuss our latest publication, and hopefully we'll get some great questions and good feedback. So just getting started, So I want to introduce nitric oxide real quick. Basically, we're a nitric oxide lab, and our paper discusses a sort of a new signaling mechanism of nitric oxide. For those of you who are not familiar with nitric oxide, this is not like most protein or steroid hormone or even other signaling molecules. It's a small free radical gas that has a very short half-life. It's synthesized biologically in most cells of our body, or a variety of different cells in our body. It's synthesized enzymatically from the enzyme nitric oxide synthase, of which there are 3 isoforms, and the cellular and tissue distribution varies based on the function and whatnot. So if we can just bring up this screenshot here. So as I mentioned, it's synthesized enzymatically. Previous slide there. Previous slide, Jason.
Yeah, that should be it.
There we go. Yeah, so there are 3 isoforms of this enzyme. You can see the neuronal, inducible, and endothelial. And the substrates for the enzyme are arginine and oxygen, and the products being nitric oxide and citrulline. And so the interesting thing about this nitric oxide is it's a small molecule. It's freely diffusible. It's not stored. It's synthesized right at the point at the location of where it's needed. Then it diffuses out and interacts with a variety of different targets. So it has a short half-life, somewhere on the order of milliseconds. And if we can go to the next slide there, I'll just want to talk briefly about what's known about nitric oxide signaling. There's 2 dominant mechanisms known of nitric oxide signaling. There's nitrosylation, which is basically the interaction of nitric oxide with metals or metal-containing proteins, primarily heme proteins such as soluble guanylate cyclase, where NO can come in, bind to the active site of these enzymes, and either activate or inactivate their catalytic activity. And then there's nitrosylation, or I'm sorry, nitrosation, which is essentially the addition of a nitrosonium equivalent to a thiol, where we have nitrosothiol formation. And this is thought to be another important aspect of NO signaling. So really, those are the 2 mechanisms of NO signaling that we focus on. But our paper really sort of highlights a new, previously unrealized mechanism of NO signaling that we're going to discuss now.
So the question is, what was the body of evidence before you guys started on this work that suggested that something like nitric oxide could be involved in epigenetics? What led you to that?
Yeah, so I mean that's an interesting question. Like most hypotheses, ideas, you never end up where you started. And so about 3 years ago, which is how long it took us to end up coming up with this paper and a lot of the studies we did, we were really interested in how nitric oxide was interacting with free iron. So one of the things that's really in all of your cells, you have a large amount of iron. Some of it's in iron sulfur cluster proteins and hemes, and but about a very small percentage, anywhere from 0.5% to 3% of your iron is floating around, and it's just there to be interacted with. And exactly what it's doing isn't always entirely clear. And so we were exposing cells to nitric oxide and examining what was going on, because it was known that this was a primary target of nitric oxide, and people had seen the formation of products with this iron as early as the 1960s, but nobody really knew what was going on, what was the biological significance of this. So we were doing a lot of interesting quantification, we were poking around on it, and we really were looking for some more physiological significance is what we were actually shooting to find. And so one of the things we started to look into is there was a lot of proteins that utilize that free iron, utilize it as a catalytic center. And there's some evidence that several of them, a couple of them anyway, are actually inhibited by nitric oxide. So once we saw that, we got excited to kind of start looking into, well, maybe there's some proteins here that nitric oxide's interacting with that haven't been realized before. And so our very simple model here was that the idea was that nitric oxide could be inhibiting some of these lysine-specific demethylases. So much like some of the other oxygenases that are inhibited by nitric oxide, these guys utilize free iron in their catalytic pocket in order to place an oxygen onto various different substrates. And in this case, there was this whole set of enzymes that were using this iron and oxygen combination in order to take the methyl group off of a histone residue, and you therefore change the methylation status of a histone. And of course, once you've done that, you've then opened up a whole possibility of regulation that isn't just through these classical signaling pathways. You have a direct inhibition of an epigenetic regulator itself. And part of the reason, if you just look at this mechanistically, that we really thought this was a viable pathway is that very similar. I mean, nitric oxide and oxygen are very similar structurally, and nitric oxide just has an extra, more of a free radical character, and actually that allows it to bind iron with a higher affinity than oxygen, and so it's actually capable of diffusing into the pocket and preventing oxygen from coming on and essentially locking the enzyme into place and keeping it from doing its thing. So that was just the idea at this point, because people had sort of shown that it could do that under a couple different settings that weren't related to epigenetics at all. And that was our opening idea, is that there's all this interaction of free iron and nitric oxide. And the thing that really kind of let us be lucky is that those lysine-specific demethylases, nobody had really been looking at them until around 2004, 2006 is when people started to isolate those things and appreciate that a cell can actually even perform a demethylation reaction, because chemically that's very challenging. And so we're kind of just sitting in the middle of all this decent information where there's new things happening, and we basically just kind of in a lucky spot and able to combine a couple of different fields and come up with a novel mechanism that helps to explain a lot of what nitric oxide is potentially doing that's been very confusing before.
So in your paper, what was your experimental rationale? How did you decide to tackle your question?
Well, so basically we started, I mean, if you kind of go through our paper, we sort of started from the simple and we built up to the more complex. So we always like to look at things sort of at the chemical level, like look at the chemical biology of nitric oxide and look at molecular adducts that might be formed, and then see how these chemical adducts, such as these dinitrosyl iron complexes, or these NO iron non-heme iron interactions, how these might actually influence enzymatic activity, and then how this enzymatic activity might ultimately influence cell phenotype, and then cell phenotype might actually be involved in disease process or normal physiology. So we start from the very small and build up to the big. And in this case, we started just by, as Jason mentioned, looking at this class of JMJC domain histone demethylases that have this non-heme iron in them. And so we hypothesized that they would be a good target for nitric oxide. And so we started very simply by just looking at a simple test tube experiment where we just took the isolated enzyme in the presence of a histone peptide fragment that was dimethylated, and in the presence of small physiologic concentrations of NO, and tried to develop this simple assay to see if nitric oxide could just inhibit the enzyme activity of this histone demethylase. And we saw that it very strongly bound to the enzyme, the iron in the enzyme, and it also barely significantly, robustly inhibited its demethylase activity. So that was really our starting point, and for there we thought, well, this is all nice test tube chemistry. Let's try to build this up. Let's see if this holds up in cells. Let's see if it inhibits these enzymes in the cells, and does the inhibition of these enzymes in the cells also result in substantial changes in histone methylation patterns.
Raj, I can't hear you.
Sorry, I forgot to unmute myself. So, were there any caveats that you had to think about when setting up your experimental design? Did you go in knowing that there are some things that you would not be able to answer with your setup?
Well, yeah, I mean, absolutely. So, whenever you're working with nitric oxide, it's a real pain. So, actually, you have to use various different tricks with nitric oxide because it disappears so quickly, and you don't actually know the concentration of it under your experimental settings a lot of times, because you have to use these donor compounds that decay at a steady rate. They have a half-life, a very predictable half-life, but if your pH shifts just a little bit, or if the temperature is different, the rate at which they're going to decay is going to change, and then you're not going to know the actual concentration of nitric oxide present under your exact situation. So you have to use a lot of different tools, and to set things up very carefully to know what the nitric oxide actual concentrations are, which is obviously important when you're setting up a kinetic assay. And so that is a real limitation. Like, for example, even though if you look at our figure 1, there's this really beautiful graph done using that mass spec data. So by MS Maldi, we really are able to show that the Kadium can pluck off one of those or two of those methyl groups there on the histone. But the thing that is, really you don't really appreciate there is that we can't show you a true kinetic because of the nature of nitric oxide, because of its production, its consumption with oxygen concentration, and that only being an endpoint assay, we really don't have a true inhibitory concentration, a Km for nitric oxide, and how well it can truly inhibit that enzyme. So at this point, a lot of those, a lot of that is just more like kind of big picture that nitric oxide can do it.
Yeah, I'll just add to that too. So as far as caveat, I think Jason really mentioned that a lot of people don't appreciate is that most of the paper is standard molecular biology techniques, Western blots and RT-PCR and this sorts of thing. But the real challenge is working with nitric oxide, because you can imagine if this is a molecule that only lasts a couple seconds, how do you treat your cells? How do you measure its concentration? How do you manipulate its concentration and duration of exposure? And so that really became the big challenge for us, and that was kind of the fun part too, that we were able to very precisely measure NO concentrations and then subsequently correlate these to specific, cell signaling events.
And on top of that, I'll even add one more. If anybody out there is actually, if we have people who are tuning in because you're actually working on this stuff and you're curious about getting into some of these, potentially some of these Jumanji domain proteins, they're very, very difficult to work with, and I don't recommend flying blind and trying to isolate them yourself. I mean, we spent a lot of time trying to get stable protein and months to a year, like really not getting the assay up and running properly. So there was also a lot of limitations with that. If getting a functional isolated Jumanji protein is not easy.
So what would you say is the main take-home message of your paper? I mean, what do you think your paper shows that advances the field of epigenetic regulation forward?
Go ahead, Doug.
Yeah, okay, so I'll start. Well, basically, I think our paper is important from two aspects. One, it's important, I think, from the perspective of the nitric oxide field, in that it really sort of, as I mentioned in the first couple slides, that there's really these two main mechanisms, known mechanisms of NO signaling, and this sort of adds a whole new layer possibility signaling mechanism in nitric oxide via its ability to directly regulate histone methylation patterns and therefore have an effect on epigenetics and not just signal transduction and cell signaling events through that mechanism. So I think it opens up some unique possibilities for people in the nitric oxide world. But also, similarly, it's also, I think, important for the epigenetic field, in that, to my knowledge, there aren't a lot of known endogenously produced small molecules that are known to be regulators of epigenetics. So from that standpoint, it's kind of interesting also. And so we kind of, overarching theme is a hybridization of these two fields where we have this free radical signaling molecule acting as an epigenetic modulator.
And go ahead.
No, why don't you finish up your thought and then I'll ask my question.
Oh, see, I was just going to add a couple, again, there's so much to say around this topic, so a couple of things that Doug didn't touch on, that, in the field right now with nitric oxide, there's some interesting things where people really are trying to, there's a big push, you might have read about the nitrosylation, so to demonstrate that NO can act more like a phosphorylating event. In other words, it goes on there so much like a kinase and then taken off by a phosphatase. But those explanations are very, very tricky, and they require a lot of complex chemistry. And so although they're clearly happening, they clearly are a portion of the signaling, this is a much simpler mechanism that might actually explain a lot of those things that people have been working very hard to find out exactly how NO is doing its thing. And this is just, we think, a much more robust, it's a more sound model for an alternative NO signaling. And then the other thing I wanted to say is that a lot of the major things that where you look at with nitric oxide, its effects can be very ambiguous. So if you're studying cancer, people have the identical model, and sometimes nitric oxide will clear the cancer, and other times it'll make it more malignant and more invasive and pervasive into the tissues. And we think that one of the reasons this might be is that this underlying epigenetic makeup. So which of these Jumanji domain proteins are being expressed? If those are differentially expressed from one tissue to the other, they might help explain why nitric oxide has one impact one time and a different impact another time.
So the question I was going to ask is that, are there any parallels in other regulatory mechanisms where say a signaling molecule akin to nitric oxide plays along with other enzymatic regulators? Are there any other examples?
Not exactly. I mean, sort of what we want to emphasize. I mean, nitric oxide is really kind of its own beast. It's really sort of unique in the terms of signaling molecules in general, just because of some of the things we've already touched upon. It's short half-life and biological and chemical properties. So I think, you know, as far as being an enzyme inhibitor, I mean, this can be common to other small molecules.
I mean, certain ways. I mean, so you could think of steroids occasionally do somewhat similar, right? So you can have agonist or antagonist, you know, type of, you know, estrogen-like compounds and things like that. And so you could have things that might diffuse in and inhibit particular areas on different enzymes. But nitric oxide really is unique. One, because it's so tiny, and the way it behaves kinetically is just really so drastically different that it, you know, it really is. It's pretty distinct as far as we know.
Another important point I think I'll just add is that, you know, nitric oxide does a lot of things. I mean, if you go to the literature, if you go to PubMed and you put in nitric oxide and X, you're going to probably find someone's found a function or correlated it to in a disease state or something like that. But the interesting thing is there's not a lot of mechanistic explanation for how nitric oxide does a lot of its pathophysiological functions, you know, aside from, as we mentioned, you know, activation of soluble guanylate cyclase, or perhaps formation of some of these other protein addicts like nitrotyrosine or nitrosyl thiols. So a lot of these currently mechanisms of NO signaling aren't sufficient to explain the vast number of phenotypic consequences subsequent to NO exposure. And so we feel this might open up another whole layer of possibilities where some of these responses might be going through this epigenetic pathway that we've uncovered here. And hopefully that will, you know, give people will pick up on this, and this will help explain, you know, maybe some unrealized functions of NO.
So how has the JBC paper informed your own research directions? Like, what are you guys working on now?
Yeah, so, you know, there's a lot of obvious directions from there. So even though we are happy with our finding, but it's very preliminary in many ways, even though it was a very extensive study. So there's so many questions that come out, actually. So when you look at, like, all of that data, what we've really shown, if you just simplify it down, let me just pull up a slide here really quick. So if you look at overall the histones that we're talking about modifying, this is, you know, here are the number of modifications that are actually present on, you know, the histone 3. So these are over like 25 sites that are capable of being modified with upwards of 100 different proteins that interact with them. And what we've done in our paper, you see this little red circle here, and then the red square at the top, that's the only portion of this that we've looked at. We've looked at one single lysine residue on one area, one methylation state. And and then we didn't even look at it at like individual areas, so individual genes, individual open reading frames. This is all global measurements. So where we're going to be going next is to really try to get into other modifications that are potentially being moved by nitric oxide, and then to look at the specific changes that are occurring that might actually lead to actual regulations of a particular pathway, individual genes that are coming up. And so we're going to go on and be doing a lot of ChIP-seq and then a lot of microarray-based studies in order to actually, you know, move this thing forward and get it out of just theory of nitric oxide inhibiting a demethylase and get it into actual functional consequence.
Right. I'll just add, yeah, basically what Ed Jason said, you know, this is just the tip of the iceberg, and so we know that NO can do this, but how does this actually transfer from the histone modification to interacting with the DNA, whether it's opening up the chromatin or forming heterochromatin, and ultimately what does this have on gene transcription and translation and cell phenotype, and can this, you know, explain certain pathological conditions or normal physiologic conditions. In addition to just looking at histone methylation marks, there's numerous other marks that, acetylation, phosphorylation, ubiquitination, these sorts of things that may directly or indirectly be influenced by the presence of nitric oxide. There's plenty to do.
Sounds like it. So I've been looking through Twitter and our Google Plus page, and it looks like we have no questions from the audience, but I know they're listening. If you do have questions, we still have a few minutes, so you can go ahead and tweet them to us at ASBMB using the Journal Club hashtag, which is #JournalClub, and we'll see if Doug and Jason can answer them. But I'm going to ask the question that I always like asking as a journalist, which is there anything we haven't talked about that we that you wish I had asked you about? Is there something we haven't touched upon that you put in your paper that you think is an important thing to keep in mind?
I mean, I could start. I mean, one thing, you know, a big. So we, in addition to what we studied in this paper, we're basically a nitric oxide lab, and we're very interested in the chemical biology of nitric oxide. And one thing we're, you know, sort of passionate about is the interaction of nitric oxide with iron, and specifically this free iron or chelatable iron pool. And we didn't have time to talk about it, but it's in the paper, that the formation of these dinitrosyl iron complexes. And these are what we believe to be one of the largest, or the largest, NO-derived adducts in a cell that's formed upon exposure to nitric oxide. But interestingly enough, very little is known about these, about their function, about their biological activity, and this sorts of thing. And they've largely been ignored in the nitric oxide community. So we hope that this paper will start to highlight the importance of the formation of these dinitrosyl iron complexes. And we think that, in addition to this one, you know, mechanism we've shown, that we think they probably do have other important physiologic functions. So
yeah,
I always like to get that out there.
And just to, you know, add on to that, and kind of more of a philosophy in general, is I think that people get way too caught up with how is this going to cure a disease, how is this, you know, relevant to, you know, something so concrete. And, you know, a lot of what this work started on, it was observation about these dinitrosyl iron complexes. Is, you know, if that's what a cell's doing, then we need to look at it. And once you start to actually make observations, you make hypotheses that might be relevant to a cell. When you try to just think out of stuff, you know, on your own, the cell just doesn't care. And you typically go down these dark roads. You try to inhibit some protein that you think is going to knock everything out, and it doesn't work a lot of times. And so I think that this work is interesting because it's just good fundamental cell biology, and it really can. I think that, you know, in general, I think the reason why so many drugs and you have such a hard time developing compounds for disease states in this day and age is that we've made a lot of the easy hits, you know, the things that are about one single mutation that have a really nice directed drug, is we've found a lot of those. And then to take things to the next level, we just are going to have to figure out the whole multitude of reactions that are going on inside the cell. And you're going to have to appreciate the fact that the iron might be changing and the redox balance might interplay with the transcriptional environment, and how much things are phosphorylated, and where the DNA is packaged, and how many cells are talking to each other, and what's the, you know, what's the microenvironment like. And so overall, I mean, I think that's just the take-home message here, is that sometimes just making observations that start off so simple really help highlighting the complexity and help to boil some things down and potentially give you some answers that you weren't looking for.
Well, I think that's a great note to end this hangout on. We don't seem to have any questions from the audience, but feel free to tweet them to us later, and we'll get them to Jason and Doug. I just wanted to remind everyone, this is an ASBMB journal club, and we hope to showcase our future authors in ASBMB publications. And the American Society for Biochemistry and Molecular Biology publishes 3 journals, which are, I can go ahead and show you the lovely slide that we have put together, which is Molecular and Cellular Proteomics, the Journal of Lipid Research, and the subject of today's hangout, which was the Journal of Biological Chemistry. I would really, really take this time to thank Doug and Jason for giving us their time and being so gracious under technical difficulties. And
We like a challenge.
Yeah, it was fun.
Thank you so much. And please check out ASBMB's website for future events such as these, and check out all the things that we do. So thank you very much, Doug and Jason, for everything you've done. And
thank you.
Yeah, really enjoyed it. Thanks a lot for putting this together, and, you know, no worries on the technical difficulties. It was a good time.
Thanks everybody for tuning in.
And we will post the link for this YouTube video about an hour or so from now, so everybody can get to watch this. So thank you very much.